So here we are on the first of probably a couple at least lectures on mitosis and M phase. Just want to run through some of the pieces and events that are going to happen. And again, the focus here is not so much that you know all these parts, because obviously that's stuff you probably have had before, and you, if I ask you questions like that, you can look them up for sure. But the main thing is just to run through this process in such a way that you'll understand some of the key points at which regulation can be put in place, because there are going to be several times during the developmental process where the cell is going to want to basically take a handle and modify it or control it so that it controls when and where cells divide and can control the developmental processes that way. All right, so last time we talked about cell division actually being two related processes, or very coordinated processes: the karyokinesis, which is going to divide the nucleus, and the cytokinesis, which will divide the cytoplasm. And so what we're going to do is focus primarily on the karyokinesis part here with mitosis, and we'll add cytokinesis in as we go along. And some of its stuff comes in about halfway through this process. So remember that karyokinesis can actually happen by two main methods: mitosis and meiosis. Mitosis is the mechanism by which we will create daughter cells that are genetically identical to the mother cell, and this would be the process by which an embryo is going to grow from a zygote over time, and of course on into a adult organism like us. As far as being able to describe the process, cell biologists have divided mitosis up into 5 continuous phases, and we'll work our way through those and just highlight a couple of the events that take place in each one of them. So most of it's summarized here in this figure we've been looking at over the course of the chapter. So cell has decided to divide, it's gone through S phase, it's done its G2 phase thing, and now time to do mitosis as part of the cell division phase, and then of course cytokinesis will wrap things up. So the 5 phases are prophase, prometaphase, metaphase, anaphase, and telophase, and basically the cell is going to work its way through dividing its nucleus, and about the time anaphase is begun, cytokinesis will begin, and cytokinesis will be the last step where the cell pinches in two and we get 2 daughter cells. Also notice in this picture the genetic identity, so the same chromosomes that are here are now in the 2 daughter cells, even after the process of separating them has taken place. Alright, so we'll start over here at the beginning, work our way through prophase and on down to telophase and cytokinesis over the next couple of lectures. Alright, so prophase is essentially the entry point for mitosis, and this and the next phase, prometaphase, are largely about getting the cell prepared to actually do the division process. So there's several things that are going to have to take place before we can divide, and we've now just come out of G2. We've essentially checked and made sure there's no damage, and we've checked to make sure that we have all the resources we need to get through this division phase. So one of the first things that's going to happen is that nuclear matrix that we talked about in the last chapter is actually going to reorganize itself and take on what we associate the X shape of mitotic chromosomes with. So just about every picture that I've shown you so far in this course of a chromosome has had that X shape to it, and you remember I showed you actually even a picture where the packaging of the DNA was taken out and the DNA spilled out and around, and you could see that X-shaped chromosome sitting in the middle, or is that X shape sitting in the chromosome. I think I've got it coming up here again, so we'll take a look at it. So it's largely the reorganization of the nuclear matrix that is responsible for chromosome condensation. We talked about the condensing proteins a little bit ago in the last lecture, and so this guy is actually going to, or these proteins are actually going to help those sister chromatids to interact with each other. And another event that's going to be taking place here with regards to chromosome is those centromeres that are found on the chromosome are going to get built up and have several proteins assemble around them into a complex structure called the kinetochore. And the kinetochore is going to be, if you remember back from early biology stuff, the centromere is where the spindle attaches. Well, it's actually the kinetochore that the spindle is going to attach to. So we're going to build this kinetochore around that centromere structure on each chromosome. Alright, so here's a figure we looked at before where you end up with your sister chromatids. So each one of these black lines would be a double helix, and of course probably has still its packaging on it as well, the nucleosomes and the solenoid fiber and so on. So these cohesins come in and attach those to each other. And then these condensins, notice what they're going to do is kind of create these loops that are going to attach or sort of break this down and pull. Basically what it's doing is it's pulling the nuclear matrix components together as it reorganizes and holds those fibers of the DNA or the chromosome to it. And here's this figure again. So basically we're talking about this fiber here at the end of mitosis, where notice how these little matrix attachment sites have attached, but now what's going to happen is these nuclear matrix is going to assume or assemble into this X shape. So this is the matrix, so we've stained for matrix here, and you can see the DNA spilling out where all the other proteins in this structure have been removed. So that's the primary thing that's happening inside the nucleus during the prophase stage. And the second thing that is going to be taking place outside, and that is going to involve the cytoskeleton that we talked about, and is going to reorganize the microtubules in the cytoskeleton into what we call the mitotic spindle. This is actually going to be a two-sided structure that is going to have essentially a star shape, and in fact it's called an aster, and each side of that is going to interact with each other. So remember that we talked about the centrosome being duplicated in S phase. Well, now basically you have two centrosomes inside the cell, and each one of those is going to begin to build one half of that spindle. And if you think about it, they're really doing what they normally do, where you remember the centrosome in a normal cell has these microtubules basically radiating out from them, and yeah, they crisscross a little bit in a normal cell. In this case, each spindle, or each half of the spindle, these centrosomes are going to radiate microtubules out from them. In this case, they're going to be a little bit more arrayed from center to out, or in to out, and what will happen is that the two are going to begin to interact with each other and push on each other. So as we kind of create these star shapes and they touch and begin to push, they'll eventually find themselves on either side of the cell. And this is largely what's going to determine what we call the polarity of the cell. Alright, so a couple of diagrams here. This is all sort of intro biology material, but just wanted to throw it in here so it would refresh your memory on some things. Chromatids condense, and this centrosome has already duplicated and now will begin to create its own little aster structures, and will begin to push on each other so that eventually they will be operating from opposite ends of the cell. Okay, so here's another way to look at it. And this would be what a normal cell looks like, and then once the duplicated centrosomes are in there, these start to build their own star shapes, and those star shapes begin to push on each other. And you'll notice the chromosomes are going to be part of this mechanism as well. So we'll talk about those here in a second. Here is a comparison of the two. So centromere here, fibers radiating out in a normal cell. Here we now have 2 centrosomes, each one in charge of a star shape, and those star shapes are going to create this spindle, we call it, with its 2 sides. Within the spindle now, there are actually several different kinds of microtubules, and they are based on the function that they have. And of course, this is the one we usually will focus on, or just whenever we talk about the spindle pulling the chromosomes apart. It's actually these guys, the kinetochore microtubules. So these are the ones that reach out and are going to attach, and both sides have got to attach to all 46 chromosomes in our cells. We have what are called the interpolar. These are the ones that are pushing on each other and are interacting with each other's, the microtubules on the other side. And this is actually going to take place through some of those motor proteins. Remember how you kind of walk along things? So if one is walking along another, then what it does is it helps those get pushed apart. So where they, once they've pushed apart and are at opposite ends of the cell, of course the midpoint is then established, and this is where our metaphase events will take place. A third kind of microtubule, and they're a little bit harder to see in the picture, are what are called the astral microtubules. They are actually on the opposite side of each star shape. So I'm going to go backwards here. So we would have kinetochores in here attaching to the chromosomes. So that would be these guys. These little pink structures are the centromeres. So kinetochore microtubules are attaching inside. You would have these other microtubules that are reaching all the way across. Those would be the interpolar. And then these out here are going to be the astral. And so notice that they're pushing across the surface. And so basically what that means is that we're kind of spreading out and pushing against the other components of the cytoskeleton, so that the cell expands as these mitotic spindles are pushing against them. Alright, so this is another figure of it, and I like this because it's actually got them labeled. So again, 2 centrosomes, and you can see these guys in place. Here's the motor proteins I was just talking about. So as they basically walk along each other, it separates the 2 of them. Here's the kinetochore microtubules attaching to the centromeres, and then these would be the astral, basically pushing out on the cell and making the cell expand as this process unfolds. So here is a cell fairly early in the process, and you can see the 2 different centrosomes. They're beginning to push on each other and will eventually be at opposite ends. The red, remember, is the microfilaments, so we'll talk about them later. You can definitely see the astral fibers pushing against those. Here the nucleus is still fairly well defined, so this is again early, but you can really see now the chromosomes starting to condense and take on those little sort of worms or snakes or whatever you want to call them inside the nucleus as the chromosomes condense. Okay? Alright, so once we've gotten past this, there's actually a couple of events before we can move forward. You'll notice here that the kinetochore microtubules are not able to attach yet, so we're not quite there, and we have to basically take this apart to be able to separate it here in a second. So that's going to be prometaphase. Alright, so the next lecture, what we'll do is we'll do prometaphase and anaphase, and then after that we'll finish it out with telophase and cytokinesis. See you then.